Okay, so let's get right into it. Deep inside every single one of your cells, tiny little machines are working nonstop, building the very proteins that make you, you. But how do they know when a protein is finished? How do they know when to stop? It's like the most important piece of punctuation in the entire book of life. And today, we're going to look at the incredible science of how that works and how we're learning to hack it. The best way to picture this whole process is to think of yourselves as these bustling, microscopic kitchens. They're constantly cooking, but instead of food, they're whipping up thousands of different kinds of proteins. These proteins do pretty much everything needed to keep you alive. This diagram here gives us a fantastic starting point. All right, so in our little protein kitchen, your DNA is the master cookbook. It's got all the recipes. When the cell needs a specific protein, it makes a photocopy of just one recipe. That's our messenger RNA, or mRNA. The ribosome is the chef who reads that recipe. And then you have the kitchen assistants. These are called transfer RNAs, or tRNAs. And each one brings a very specific ingredient, an amino acid, over to the chef. So you've got the chef reading the recipe and the assistants bringing ingredients one after another, building this long chain that will become a protein. But every good recipe has to have a final step, right? It has to say the end. If it didn't, you just get this long, jumbled mess, a protein that's totally useless and could even be toxic. Now, the language of that mRNA recipe is written in these three-letter words called codons. And it turns out there are three specific codons that just scream stop. They are UAA, UAG, and UGA. Whenever our ribosome chef gets to one of these, it knows the protein is done. So here's where it gets really interesting. You'd probably guess that another kitchen assistant, another tRNA, shows up, maybe carrying some special end ingredient, but that's not what happens at all. For this one final, super important job, the cell calls in a completely different kind of molecule. And the molecules that answer that call are a special group of proteins called release factors. In organisms like us, complex ones, the main player here is a protein called ERF1. So let's go back to our kitchen. If the tRNAs are the assistants bringing ingredients, the release factor is kind of like the kitchen supervisor. And it's totally different. First off, it's a protein, not an RNA molecule. And it's not carrying an ingredient, an amino acid. Its one and only job is to see that stop sign and shut the whole production line down. And this is where nature pulls off one of its cleverest tricks. It's called molecular mimicry. The release factor protein has evolved to be shaped almost exactly like a tRNA molecule. It's basically wearing a disguise. This lets it slide right into the spot that's reserved for the next kitchen assistant, totally fooling the ribosome. Okay, so the supervisor in disguise has docked. It's fooled the chef, and production has ground to a halt. But there's one last piece of business. The brand new protein is still stuck. It's physically attached to the last tRNA. So how does the release factor actually cut it free? This is where the story just got a major update, flipping a long-held theory on its head. For decades, scientists figured that release factors were like a pair of molecular scissors, that they actively cut the protein chain. But as this research shows, the truth is way more elegant and, frankly, way cooler. It turns out the release factor doesn't do the cutting itself. Instead, when it docks, it subtly changes the shape of the ribosome's machinery. This little nudge pushes the end of the new protein into a weird, unstable position, basically forcing the protein to sever its own bond to the tRNA. It's a process called autohydrolysis, a self-cut. The supervisor doesn't fire the worker, it makes the worker quit. It's an unbelievably clever and efficient system. But it all depends on that stop codon showing up in the right place. So what happens when a random genetic typo, a mutation, creates a stop sign smack in the middle of a recipe? Well, that creates what's known as a premature termination codon, or a PTC. The ribosome, it's just doing its job. It sees the stop sign, calls in the release factor, and bam, production stops. The result is a shortened, chopped-off protein that is almost always completely broken and useless to the cell. And this isn't some super rare thing.
These kinds of mutations, called nonsense mutations, are thought to be the cause of about 12% of all inherited human diseases. That's a huge number, affecting millions of people all over the world.
This one type of molecular error is the root cause for thousands of genetic disorders. We're talking about devastating conditions like Duchenne's muscular dystrophy, which causes muscles to waste away, and a significant number of cases of cystic fibrosis. But here's the amazing part. By understanding this process at the deepest, most fundamental level, how the release factor binds, how it fakes out the ribosome, how it triggers that final cut, scientists can now start to develop some really ingenious ways to step in. They are learning how to hack the stop codon.
So the first big idea is something called read-through. The strategy is to design a drug that basically tells the ribosome to run the red light, to just ignore a faulty, premature stop codon. These drugs work by getting in the way of the release factor, so it can't bind properly. This gives a normal tRNA kitchen assistant a chance to sneak in with its amino acid, and the ribosome just keeps on building the full-length working protein.
So how in the world do you find a drug that can do that? Well, you use really clever screening tools, like the one you see here. What they do is they set up a test tube with ribosomes that are stuck at a stop codon. When they add the release factors, the protein gets cut and a fluorescent signal plummets, just like you see on that graph. But if they add a drug that actually works, it blocks the release factor and that signal stays high. So they can screen thousands and thousands of chemicals to find the ones that keep the light on, pointing them toward a potential new medicine. But the ultimate hack? Well, it goes even further. Instead of just trying to get the ribosome to ignore a bad stop codon, what if we could actually change what the stop codon means entirely? What if we could actually go in and rewrite the rulebook of the genetic code?
This is not science fiction. This is what synthetic biologists are doing right now. In a groundbreaking experiment, scientists went through the entire genome of an E. coli bacterium, and they swapped out every single instance of one stop codon, TAG, and replaced it with another, TAA. Then, they just deleted the gene for the release factor that recognized TAG. All of a sudden, the TAG codon meant nothing. It was a blank slate. They could then add a brand new, engineered tRNA and program it to insert a synthetic amino acid, one that doesn't exist in nature, opening up a whole new world of protein design.
So from developing drugs that can fix a genetic typo to completely reprogramming life itself to build things we've never seen before, we are really entering a new era. It shows us that in biology, that stop signal isn't always the end of the story. Sometimes it's the gateway to a whole new beginning.